CECAD is the Cluster of Excellence for Cellular Stress Responses and Aging-Associated Diseases. And what we're doing at CECAD is a wide variety of aging research. Cellular stress responses are really at the heart of what is happening in the aging process.
The greatest risk for impairment of the regenerative capacity is aging. So basically we see problems of skin repair and chronic wounds, dry skin, barrier defects, particularly in the elderly. Here it would be fantastic and a great help to better understand the underlying molecular mechanisms, and then also to be capable to develop maybe specific therapeutics and approaches to address those problems.
In research area 2, we are interested in understanding how different organs interact with each other in the aging body. We know that organs are constantly in communication with each other. The brain communicates with the intestine, the intestine can communicate with the germline, and different tissues interact with each other.
In certain aspects, we have skin diseases that are limited to the skin. There are pure, clear skin diseases. But on the other side, we have also skin diseases, which we know which go very much together with other diseases.
Now in the human, that's a very complex process. For example, what we are interested in is how the stability of the genome is maintained and how DNA damage in every single cell drives the aging process. They are humans, children that age within their first decade of life. And they are born with defects in genes that are necessary for repairing the DNA in all of their cells. And normally for a number of decades, until our midlife, our cells repair quite well, but then damage accumulates just like in these patients and leads to aging-associated disease, to the functional decline that we experience when we age. These human syndromes are extraordinarily complex disorders, and that's why we study them in a very simple nematode worm, so we can understand how these repair mechanisms operate, how the organism, the developing as well as the aging organism, responds to the damage that cannot be repaired and then leads to the accelerated aging. And what we discovered was that the entire animal responds to that, to this very specific DNA damage in germ cells, and it elevates its resistance to all kind of stress. And this endurance that the animal gains can actually extend the reproductive lifespan of this animal and allow the germ cells to repair their genomes and then produce offsprings later in its life. We also discovered that there are mechanisms that impinge on every single process in a cell that responds to DNA damage, and they can actually extend lifespan under the influence of DNA damage. And it impinges so many different processes that go awry during the aging process that we can identify now new targets that could enhance the functionality of cells and tissues despite the invariably accumulation of DNA damage during our lifespan. So for our research, it is very important that we can use state-of-the-art technology. We can study the cellular processes by investigating every single protein and every single gene of the animal. We can in detail analyze what is happening during the aging process, what is happening when animals respond to the accumulation of DNA damage.
We have a wound healing clinic where we see, for example, and this is one of my main interests also, where we see, for example, patients with impaired wound healing conditions. Impaired wound healing conditions are usually, when you see them at the skin, they are also the cause of an underlying systemic disease or condition as is aging. So most of our patients are old patients. So basically there we have already a rich source of selected patients which we can study. We observe, we categorize, we take the history, we document the patients, the skin defects and also the underlying systemic diseases, and We take tissue samples, serum samples from those patients, which are clinically then very well characterized. In addition, in the laboratory, we also use animal models. Here we use mostly mouse models, where we ask very specific questions and then have a systematic analysis also on the molecular analysis. The findings are that there are conserved mechanisms between these different model organisms and the patients. So one example is the target of rapamycin pathway. So the TOR pathway is considered currently as one of many other central pathways that controls aging. And what we observe, for example, now in the skin is very similar in the mouse from what we see also in patients. So there are similarities, and that means that we can use and compare also these different models. Over the past 10 years, we had a major development in dermatology. We have new therapeutics on all different levels, which also, maybe not directly, but indirectly, maybe impact regenerative capacity and thereby also indirectly might impact aging processes. So if we maybe repurpose medications that are now new on the market and that we can use maybe also to address pathways and mechanisms that we identify that are altered in the aging body and organism, and that we could then maybe use also in the clinic.
Aging is a complex process. There's not one super pill that reverses aging on itself, but instead we need to understand the complexity of aging. We need basic research to really understand what are the processes of aging and how are they connected to each other. And only then will we be able to really solve a major problem of society, which is that we have a large proportion and an increasing proportion of our population suffering from chronic diseases.